You cut your finger. It's small, barely anything. You hold it up and watch, and something thick and dark wells up from the skin. Not water, not a clear trickle. Something red, something slow, something that within minutes begins to seal itself shut like it was never broken. You are 70% water. You have been told this your entire life. So why, when something punctures you, does the ocean inside you not just pour out? That question has a real answer. Actually, it has 6, and each one is stranger than the last. Let's go. Here's the first thing almost nobody gets right. People assume the body holds water the way a sponge does, loosely, passively, ready to drip at the slightest pressure. That's not even close to what's happening. The body doesn't hold water in one place. It holds water in at least 6 structurally distinct compartments: intracellular fluid, interstitial fluid, plasma, lymph, cerebrospinal fluid, synovial fluid, each maintained at different pressures, different chemical compositions, separated by membranes that actively pump ions to maintain those differences. Water inside you is not resting. It is being managed, moved, sorted, and locked into place every second of your life. Peter Agree at Johns Hopkins won the 2003 Nobel Prize in Chemistry for discovering aquaporin's protein channels embedded in cell membranes that control exactly where and how fast water moves through tissue. A single red blood cell contains roughly 200,000 of these channels, each one moving water at 3 billion molecules per second. That is not passive. That is architecture. Your cornea holds exactly 78% water drift, 1% in either direction, and your vision distorts. The inner core of each spinal disc is 80 to 85% water, held under 3 atmospheres of pressure by a fibrous outer ring. Knee cartilage traps water in a pressurized gel matrix, using molecules called proteoglycans, with almost no blood supply involved at all. The body is not a water balloon. It is a pressurized city, and every district has its own plumbing. This system evolved because single-celled ancestors lived in open water where osmotic pressure was constantly trying to either flood or drain them. Compartmentalization was survival. Over 500 million years, that survival mechanism became the architecture of the entire vertebrate body. The body isn't full of water. It's full of decisions about where water is allowed to go. Now, let's talk about the red part. Most people think blood is basically red water, a simple liquid carrying oxygen. What blood actually is: a suspension of living cells inside a protein-rich fluid so complex that a 2006 Stanford study by Alexei Nikitin and colleagues identified 1,444 distinct proteins in human plasma alone. Earlier estimates had placed the number near 490. The gap between what scientists assumed and what was actually there was staggering. Blood is not a fluid. It is closer to a mobile organ. The red cells, which give blood its color, make up only 40 to 45% of its volume. And hemoglobin, the molecule doing the oxygen work, didn't even evolve for that purpose. It evolved roughly 800 million years ago as an oxygen scavenger, a molecule that protected early anaerobic cells from toxic oxygen before being repurposed for delivery. The version inside you is not the original design. Horseshoe crabs use hemocyanin instead, copper-based, which turns blood blue when oxygenated. And their clotting proteins are so sensitive to bacterial toxins that every FDA-approved injectable drug on the planet is still tested using them. Antarctic icefish have no hemoglobin at all. Their blood is colorless. Oxygen dissolves directly into plasma at near-freezing temperatures, a discovery Johann Roode made in 1954 that rewrote what scientists thought blood had to be. Red blood isn't the standard. It's just the version that won. But here's what nobody tells you. All of this containment, all of this engineering, all of this 500 million year plumbing has exactly one weak point. And for most of human history, that one weak point killed kings. When you do break through the skin, something immediately begins that most people misunderstand completely. Blood doesn't clot because it dries. It clots because it builds a structure. A biochemical cascade, at least 13 protein factors, labeled I through 13, activates in precise sequence like a molecular relay race. Each step amplifies the signal. One molecule of factor XA generates 1,000 molecules of thrombin. Fibrinogen, a soluble protein floating in your plasma, converts to solid fibrin threads within 15 to 30 seconds of injury, forming a mesh that catches platelets the way a net catches fish. The whole process requires specific membrane surfaces, specific cofactors, specific chemical conditions. The complexity isn't an accident, it's a failsafe. The cascade is deliberately hard to trigger, so it doesn't fire inside undamaged vessels. Harold Macfarlane at Oxford and Earl Davey at the University of Washington published this model simultaneously and independently in 1964, one in Times Nature, one in Times, Science Times, both reaching the same conclusion through different work. NASA research published in 2016 found that in microgravity aboard the ISS, clot formation slows measurably, suggesting gravity helps platelets aggregate in ways we still don't fully understand. In the earliest open-heart surgeries, bypass machines sheared platelets so aggressively they triggered partial cascade activation, causing microclots to form throughout the body. The system requires the right conditions, in the right order, for a reason. Your blood doesn't clot because it hardens. It clots because it builds a wall, on purpose, in under 4 minutes. And the vessel itself is not passive either. The endothelium, the single-cell thick lining of every blood vessel in your body, actively secretes at least 12 identified signaling molecules that tell blood whether to clot, whether the vessel should dilate or constrict, whether to allow immune cells through or block them. Robert Furchgott, Louis Ignarro, and Farid Murad won the 1998 Nobel Prize in Physiology or Medicine for discovering that endothelial cells produce nitric oxide, a gas that signals surrounding muscle to relax and widen the vessel. Furchgott's original 1980 experiment proved this by removing the endothelium entirely, at which point the vessel constricted instead of dilating. The lining had been producing a constant relaxing signal. Take it away, and the pipe collapses. During exercise, endothelial cells in working muscle detect the shear stress of increased blood flow and release nitric oxide within seconds, dilating those vessels by up to 30% while constricting vessels elsewhere. In sepsis, bacterial toxins destroy endothelial function, and vessels that once held fluid begin allowing plasma to pour into surrounding tissue, the leaky capillary syndrome that kills septic patients. Not the bacteria. The breakdown of the gatekeeper. The placenta builds an entirely new endothelial network within weeks of conception, and the fetus chemically signals the mother's vessels to dilate through that layer, essentially hijacking maternal circulation. The wall of your blood vessel isn't keeping blood in. It's constantly deciding whether to let it out. Now, for the exception, that proves everything. Hemophilia, the inherited absence of a single clotting factor, typically factor 8 or 9, renders the entire coagulation cascade non-functional. A person with severe hemophilia A has less than 1% of normal factor 8 activity. Minor injuries bleed internally for days. Joint bleeds destroy cartilage progressively. Without treatment, historical life expectancy was under 20 years. Alexei Romanov, Tsarevich of Russia, born 1904, inherited hemophilia B from his great-grandmother, Queen Victoria, a spontaneous mutation confirmed by DNA analysis of Romanov, remains by Evgeny Rogayev at the University of Massachusetts in 2009. Alexei's condition was the gravitational center of his parents' desperate reliance on Rasputin, which directly eroded the dynasty's credibility in the years before 1917. Queen Victoria's son, Leopold, Duke of Albany, died in 1884 at age 30 from a cerebral hemorrhage after a minor fall, an injury any non-hemophiliac would walk away from. One missing protein, one broken step in a 13-step cascade. One empire destabilized. The complexity of the coagulation cascade is not over-engineering. Every single factor is load-bearing. There's one last thing I saved for the end. The question you started with, why doesn't the body just leak, is not actually a biology question. It is a physics question. Physics wants your water back. The second law of thermodynamics describes the universe moving toward dissolution, toward equilibrium, toward the loss of organized structure. Your body is a temporary, energetically expensive rebellion against that. Every compartment, every clotting factor, Every endothelial gate, every aquaporin channel, all of it is life saying no to entropy. The kidney filters 180 liters of your blood every day and returns 99% of it. Homer Smith traced the kidney's evolutionary history in 1953 and showed that the glomerulus, the kidney's filtering unit, first evolved in freshwater fish facing osmotic dilution 400 million years ago. You are still running that fish's plumbing. It has just been scaled up, layered over, and kept running for the entire history of vertebrate life. We built modern medicine on understanding why blood stops. Carl Landsteiner's discovery of blood types in 1901 made transfusion possible. The synthesis of recombinant factor VIII by Genentech in 1984 saved hemophiliacs, though it arrived too late for the 30,000 people infected with HIV through contaminated blood products in the prior decade. The nitric oxide discovery led directly to sildenafil, originally a cardiovascular drug, repurposed after its vascular effects surprised everyone in clinical trials. Every surgery ever performed depends on the same molecular cascade that evolved in jawless fish 450 million years ago. Modern medicine is not separate from evolution. It is evolution, looking at itself and finally understanding what it built. Go back to that cut, that small one on your finger. The red that appeared wasn't a failure of containment. It was containment thick, protein dense, engineered across 500 million years, already building its wall before you'd finished watching it form. Your body is 70% water. It just built a city around that water, pressurized every district, installed 13-factor security at every exit, and spent 500 million years making sure it never, ever just leaks. You are mostly water. You just happen to be water that decided to stay.